Hello, my name is Jackson Cooper, and my project is evaluating the effect of copper acclimation on the efficiency of the eastern oyster at filtering nitrates from eutrophic or nitrate-rich water. So the central problem fueling this research is eutrophication, and eutrophication is the process by which nutrients from land sources like fertilizers are washed into nearby bodies of water, and these fertilizers contain high concentrations of nitrates and phosphates. And when they enter the marine environment, they're good for terrestrial environments, of course. Farmers use them to prevent or promote plant growth. But like any good thing, too much is dangerous. So when it gets into marine environments, an algae called Karenia brevis, which is the algae that is responsible for red tide in the Sarasota Bay area, it outcompetes other algae in open water, and it floats into nearby bodies of water, like, for example, Sarasota Bay, where excess nitrates and phosphates are becoming present from leaching nutrients from the soil when rain comes down and washes them into these nearby bodies of water. And when these algae start to use all these excess nutrients to grow, they form these giant blooms of algae or blankets on the surface of the water. And these blooms are so large and dense, they block light from penetrating the water column and prevents other marine plants at the bottom from absorbing light and performing photosynthesis. So significantly less energy is produced by marine plants in the affected areas. And the algae blooms eventually run out of nutrients when they grow too much, and the algae begins to die and sink to the bottom of the ocean to decompose. But the decomposition process is done by bacteria, which requires a lot of oxygen. And when the oxygen level is depleted, many sessile or attached organisms like oysters, which we'll be using in this project, and mussels, they begin to quickly die, and many organisms have to migrate out of the area to avoid death because they can't get any oxygen to perform respiration. So, many organisms don't make it out on time, however, and beaches begin to see thousands and thousands of dead fish washing up on the shore. Of course, we see this in Sarasota almost year-round, it seems. And of course, no tourist wants to go sit on the beautiful beach after it's been converted into basically a marine morgue. So, the particular species of algae responsible for red tide in Sarasota Bay produces a harmful neurotoxin called brevetoxins. And this is the chemical layout of a brevetoxin. And brevetoxin begins to build up in cleaner fish that eat producers like the algae that float into our bays. And as it enters their bodies, it builds up and affects the brain and eventually causes them to die. That's how you see those big clumps of fish ending up on the beach. And not just affecting single species of fish, of course, it's going to affect the entire food web. So as brevetoxins build up in one species, another species eats them, it's going to build up in their tissues, and those concentrations move up the food web. And it's a process called biomagnification, where the concentration of brevetoxins are going to increase as they're moving up the food web, being eaten by animals in higher trophic levels than them. Now, brevetoxins are not only dangerous to marine organisms, they're also very dangerous to humans. So since it is a neurotoxin, if humans breathe in these brevetoxins, they're often aerosolized by water movement. They can affect the brain, of course, it's neurotoxin, which can cause respiratory issues, problems with the stomach and intestines, and it can cause a lack of muscle control and coordination. So there's a lot of different issues that can go into human being affected by these poisons. So what have humans done about this so far? Scientists have been working with bivalves. It's a class of shellfish that comprises of organisms that are filter feeders, like these clams here. So they clean a wide variety of macro and micronutrients from the water. And in a Stanford study, clams and mussels were found to be extremely efficient filter feeders, able to remove 80% of waterborne chemicals in a very short time frame. So they've already proved themselves as useful tools to clean our water. Now, many studies have been done on clams and mussels, but this research focuses on the delectable shellfish treat known as oysters. And the eastern oyster is a native species in the Gulf of Mexico and in Sarasota Bay, making it the perfect candidate for this study. So oysters are one of the most important bivalve filter feeders we have on the planet. They, similar to clams and mussels, suck in water and remove chemicals and nutrients suspended in it. They use many of the nutrients for growth, but the excess turns into waste that many other species rely on for food. And one adult oyster can filter up to 50 gallons of water in a single day. So clearly they play a key role in the health of the marine environment. How? First, they reduce water turbidity by removing large suspended particles. Second, the most important part of this project, they removed nitrates from the water that algae can no longer use to create harmful algal blooms. Third, they can be the base of artificial reefs. So instead of rock-based reefs where coral polyps attach to the rocks, oysters provide that solid, sturdy surface for coral polyps to anchor down and create those beautiful ecosystems that we see. And finally, they help with erosion prevention. So those solid shells that they have, they act as a breakwater. So they take away the energy from the moving water, and it prevents the coast from being washed away by strong waves. So there have been previous studies that show filtration power of oysters, but new variables haven't really been introduced much. So this leads to our project's main variant, which is copper. And copper is a heavy metal found in trace concentrations in seawater, only about 0.09 parts per million. So for every million parts of water, there's only 0.09 parts copper. And however, this is an extremely important metal for oysters. Even though it's a heavy metal, it can be dangerous in high concentrations, and oysters use it for filtration and metabolic processes. So like I said, too much of a good thing is dangerous, of course, with heavy metals. If the concentration of copper in seawater is too high, it can cause several issues in fish species. So copper can disrupt gills, making fish unable to regulate their salt levels, often causing the fish to dry out. So if they build up too much salt in their tissues, they lose more water and they eventually dry out. It also disrupts the smell of fish, which goes hand in hand with their sense of migration, communication, and smell. So all these things led me to my hypothesis. If the concentration of copper in an acclimation tank is increased, so we put the oysters in a different tank, because if you dump water out into the marine environment, it's going to have all those negative effects on fish. So if copper increases in a separate tank where just oysters are able to utilize those increased levels of copper, then nitrate concentration will be decreased when the oysters are used to filter out those nitrates in test tanks. So to conduct this experiment, I gathered 4 of these beautiful 10-gallon tanks that were filled with water from Sarasota Bay, just to ensure that the oysters weren't introduced into water that they weren't familiar with and had different chemical levels and such. And there was also an experimental tank, which was just a 5.5-gallon tank that was also filled with the same seawater. The 10-gallon tanks were filled with sand for the oysters to rest on, but the 5.5-gallon tank did not have sand because it was hypothesized that the sand would absorb nitrates and possibly skew the results. Every tank was outfitted with filters, bubblers, temperature regulators, keep the water conditions stable for the oysters so they're not seeing too much of a change in chemical levels. And chemical levels for nitrates, nitrites, and ammonia, those are just 3 standard nitrogen compounds that most aquarists look at to make sure their organisms are in safe water conditions. So those are measured periodically in the acclimation tanks, and also salinity was tested too, because oysters cannot easily regulate their salinity. All right, and then finally the oysters were added, about 6 per tank. And they were set for about a week or 2 just so they could get all that copper moving throughout their systems, accumulate it in their tissues, and begin to use it in their filtration processes. So for the testing phase, 1 group was moved at a time and left for 2.5 hours. The water was sampled every 30 minutes, and this was resulted in 6 samples. So we have the original sample plus 5 test samples. And this was taken from information that stated that oysters were able to filter 2 gallons of water per hour. So in 2.5 hours, they should be able to filter all of the water in that tank, hypothetically. Finally, these samples were ran through a colorimeter using the given instructions. So what we saw in the 0.75 concentration tank was an overall increase, which was the exact opposite of what was expected. The same thing occurred in the 0.25 part per million group, although, as you can see, from 13 about 0.258 to only about 13.5 was barely an increase at all, so not much happened in that group. And the control group, barely saw any of an increase at all. It was almost completely flattened out by about half an hour. The only group that showed a somewhat supported the hypothesis was the 0.5 part per million group. It actually showed an overall decrease, which was what I hypothesized would happen. There were a few points in the graph about 2 hours in where the nitrate level did actually increase, but it's possible that could have been through waste excretion or other variables. So this shows all of the groups together. As you can see, the only group that followed the hypothesis and showed a decrease in nitrate concentration was the 0.5 group. 0.75 group almost doubled, or almost tripled actually, and the 0.25 and 0.09 groups changed so small that you can barely see them on the graph. So if we go back to our hypothesis, that increased copper concentration in the acclimation tanks would result in decreased nitrate concentration in experimental tanks. The majority of the groups did not support the hypothesis, with the exception of the 0.5 part per million group. So a one-way ANOVA test or analysis of variance was conducted on nitrate concentrations and also percent change in nitrate concentrations. So for the nitrate concentration group, when the ANOVA test was conducted, the p-value was extremely small, which I was surprised at since barely anything happened in 2 of the tanks, and in one of them, the complete opposite of what I expected happened. But the fact that nothing happened in 2 of the tanks could be the reason that the p-value was so incredibly low. And when I conducted the same test on the percent change in nitrate concentration group, the p-value was also very small, but not as small as the nitrate concentration group. But both groups showed significance, although I question this just because of how the experiment went, with barely anything happening in 2 of the tanks and the exact opposite of what I expected happening in 1. So I question the significance of the data. And if there was one thing that was significant, obviously, it was the number of sources of error, let's be real. So I believe the SMART-3 colorimeter that I used to test the samples provided a noticeable source of error. It's always possible that small, almost unnoticeable differences in the test procedure could have messed up the results, even though I followed the instruction they gave almost to a T, as close as I could. The second thing, I could not determine a pattern for times when the oysters would be feeding, so it's impossible to know if they were even filtering while they were in the experimental tanks. So during the process, it's hard to know whether they're actually going to be doing what I need them to do. So it's probably the largest source of error. And finally, was our test kit here. Since these only give a color approximation, it's difficult to know what the actual chemical concentrations were in the acclimation tanks. So it's difficult to know if the oysters are being stressed out by ammonia nitrates or nitrites in their test tanks before they went into the experimental tank. So it's possible that they, if they were stressed out, they could have performed less than what they would have done in normal chemical levels. So there are ways that we can apply this experiment to a practical setting. So in Sarasota Bay, oysters could be removed from the bay. They could be placed in these tanks, or they could be acclimated to copper at 0.5 parts per million, because that's the only one that actually supported the hypothesis and showed a decrease in nitrate concentration. And this process could continue. So taking the oysters out of the copper tanks, putting them into the bay, and then repeating the process. Of course, we'd have to keep them in separate tanks, containment, so that local species could not come in and eat them, because then we would see what we saw earlier, the biomagnification of copper this time. And we saw that it could affect fish's migratory sense of smell, salt regulation, all that stuff. So we'd have to make sure that the oysters are in separate containers to ensure the safety of other local species in the marine habitat. And by doing this, we can reduce the concentration of nitrates in our bodies of water. So back to our main purpose, preventing algae from being able to produce these large blooms on the surface of the water that cause red tide and kill so many fish every year. So in the future, I'd like to investigate the effects of other metals on oyster filtration performance, such as iron, nickel, and silver. I just believe that testing other heavy metals in the marine environment might be key to improving bivalve filtration. And I do believe that this project could provide a key way to start to reverse the effects of red tide that we see every year. Yeah, thank you, Tiha.